Let's start by understanding how our immune system works in two main phases. First, the innate immunity acts quickly within hours to provide a general defense, using barriers like skin, phagocytes that engulf invaders, complement proteins, and natural killer cells. Then, over a few days, the adaptive immunity kicks in, where B cells produce antibodies and T cells become effectors to target specific threats. This timeline shows how these two systems coordinate to protect us efficiently. Phagocytosis is a key process where certain immune cells engulf and destroy harmful microbes. It begins with the immune cell moving toward the microbe and sticking to it. Then, the microbe is ingested into a vesicle called a phagosome, which fuses with a lysosome containing enzymes. These enzymes digest the microbe, leaving behind waste that the cell then expels. This step-by-step process is crucial for clearing infections and initiating further immune responses. Phagocytosis does a lot more than just eating microbes. It's nonspecific, meaning it acts quickly against many invaders as part of innate immunity. It helps eliminate pathogens, clears out dead cells, and even presents pieces of microbes to other immune cells to trigger a more targeted response. Plus, it plays a role in regulating inflammation and activating the adaptive immune system, making it a vital bridge between early and later defense mechanisms. Now let's explore how T cells and B cells work together. When a macrophage processes an antigen, it presents fragments to helper T cells. These activated T cells then stimulate B cells to produce antibodies. Meanwhile, cytotoxic T cells target infected cells directly. This collaboration leads to two types of immunity: cell-mediated, which kills infected cells, and humoral, which involves antibodies circulating to neutralize pathogens. Memory B cells also form to improve future responses. This cycle shows how T and B cells interact dynamically during an immune response. It starts with antigen presentation to T cells, which recognize and activate helper T cells. These helpers release cytokines to signal other cells, activating cytotoxic T cells and B cells. B cells then differentiate into plasma cells, producing antibodies and memory cells for long-term protection. Regulatory T cells help keep this response balanced, preventing overactivity. This cycle ensures a coordinated and effective defense. Adaptive immunity unfolds in distinct phases over days. Initially, antigen recognition triggers lymphocyte activation, where naive T and B cells proliferate and differentiate into effector cells like antibody producers or killer T cells. This leads to antigen elimination through humoral and cell-mediated pathways. After clearing the threat, the immune system contracts by removing excess cells, maintaining homeostasis. Finally, memory cells persist, ready to respond faster if the same antigen appears again. Looking at humoral responses over weeks, we see how the body reacts to different antigens. The primary response to an antigen involves naive B cells becoming plasma and memory cells, producing antibodies slowly. Upon a second exposure, the secondary response is faster and stronger due to memory cells, especially for the first antigen. This pattern highlights how immunological memory enhances protection, enabling quicker and more robust antibody production upon re-exposure. The complement system is a group of about 30 plasma proteins that play a crucial role in defending the host. They activate in a sequence through three pathways: the classic, lectin, and alternative pathways. Each pathway triggers a cascade that helps eliminate pathogens, making complement a powerful component of our immune defense. Activation of the complement system depends on different triggers for each pathway. The classic pathway requires antigen-antibody complexes, specifically IgM or IgG. The alternative pathway is part of innate immunity and activates directly by bacterial components without needing antibodies. The lectin pathway kicks in when mannose-binding lectin attaches to microbial surfaces. These distinct activation conditions allow the complement system to respond flexibly to various threats. Here we see how the three complement pathways, classical, lectin, and alternative, converge to activate the complement system. This activation leads to recruitment of inflammatory cells, marking pathogens for destruction through opsonization and directly killing them. This coordinated response amplifies immune defense, ensuring pathogens are efficiently targeted and removed. The complement system can lead to several outcomes. It helps in bacterial lysis and amplifies inflammation to fight infections. However, it can also contribute to hypersensitivity reactions and endotoxic shock if uncontrolled. Additionally, it assists in immune adherence and opsonization, but may be involved in autoimmune disorders. Understanding these effects is important for appreciating both the benefits and risks of complement activation. Cytokines are signaling proteins that regulate immune responses by acting on the same cell that secretes them, nearby cells or distant cells. They include various types like interleukins, interferons, tumor necrosis factors, colony-stimulating factors, and transforming growth factors. Each plays a specific role in coordinating immune activity, making cytokines essential messengers in maintaining immune balance and defense. Finally, let's touch on some important immunological concepts. The MHC complex and HLA typing are key for immune recognition. Immune surveillance helps detect abnormal cells. Booster vaccine doses enhance immunity by reinforcing immune memory. Immune tolerance prevents the body from attacking itself. Together, these elements form the foundation for understanding immune system function and its clinical applications.